So, welcome to all of you from Europe, America, Middle East, and Asia. I am Norbert Claude Gorin from the Department of Hematology and Cell Therapy at Hôpital Saint-Antoine in Paris, and also I'm the chair of the EBMT Global Committee. I'm pleased, I am honored to be the moderator for this seminar dedicated to COVID. You may say again, but in fact, we are trying to cover fields that have not been covered recently. This is my disclosure, and I am pleased to mention that this webinar is supported by an unrestricted educational grant from Airinspace. We have 3 worldwide renowned speakers, and I'm grateful they accepted to participate. The first one is Jean-Philippe Desli from Commissariat à l'Énergie Atomique, and he is an expert on prevention of contaminations between people. He has worked very much on prions, but he will cover not only prions, but also SARS-CoV-2. The second speaker is Bruno Canard from Marseille, and he is an expert on drug discovery and development of therapies against SARS-CoV-2, the disease, not vaccination, but treatment of the disease. And the third speaker is Patrick Berche from Hôpital Necker in Paris and Institut Pasteur in Lille, and he will review for us the various vaccines and try to find which advantages and maybe caveats some of them may have. Please do not leave immediately after the last talk, because we will have at the end a general debate. I would like now to introduce this seminar with a few slides. First of all, here is our enemy, the SARS-2 virus. This picture that everyone knows nowadays is probably the picture that has most circulated in the world in 2020, and it's much more famous than any politician, movie star, or football player. If we look at the statistics of the Johns Hopkins Center in Baltimore, in September 2020, 4 months ago, we had almost 30 million people infected and 1 million dead, with a crude fatality rate of about 3%. But if you look now, 4 months later, now precisely, we have 90 million people infected and almost 2 million dead, with a crude fatality rate of 2.2%, which is enormous. To introduce the speech by Jean-Philippe Desli, let me show you the results of a recent study with laser light scattering of speech-generated oral fluid droplets, as recently published in the New England Journal of Medicine. You can see the flash count during each frame of a video when a speaker talks, with and without a damp mask covering his mouth. Green indicates spoken words. The number of flashes was highest, and you can see the red arrow, when the TH sound in the word healthy, so difficult to pronounce in English when it is not your mother tongue, was precisely used. The brown curve below corresponds to the situation when the speaker's mouth is covered with a damp mask, showing the importance, as we all know, of the mask. And here you can see precisely the droplets staying in the air. So while the virus itself is too small to be stopped by filters, on the other hand, equipment like the plasmare, as indicated here in China, in Suzhou, not far from Shanghai, blocks the droplets and further also destroys particles of all kinds, including bacteria, fungi, and viruses in an oxygen plasma-built environment. And here in Shanghai, in Regine, biocare rooms with negative pressure in the room to protect the medical and the nursing staff taking care of the patients. For our second speaker, a major topic at a time when everyone is concerned by the vaccine is whether we will find in the end, therapeutic tools to treat the disease itself. I took this slide from the presentation of Bruno Canard himself. We all know that many molecules that gave once hope, in fact, do not work. For instance, remdesivir, lopinavir, ritonavir, sofosbuvir, hydroxychloroquine, and so maybe Bruno Canard will find something else and will be more optimistic than I am at the moment. A few months ago, I discovered by pure chance on the web a molecule, opaganib, which combined with remdesivir in vitro was considered synergistic in destroying the virus. But so far, nobody speaks again of this molecule, and I have no idea whether the trials which are going on are going to be positive or not. Finally, for our last speaker, Patrick Berche, I have 3 introductory slides. The first one that I also took from a review Patrick Berche himself wrote recently shows us again, as we know, that we will soon have numerous vaccines with different properties, and Patrick Berche probably will explain us the different properties. But this second slide, in my view, is very interesting because it addresses an important question whether we can use a single dose administration of intranasal vaccine. And this is a trial ongoing with the Oxford AstraZeneca vaccine. And this will be probably a question for Patrick Berche because the protection might be much more important with production at the local level of IgA immunoglobulin. And the third one is this slide from the CDC. It's the first report on allergic reactions following the Pfizer vaccine. As you know, some people are concerned, some people are anxious, but as you can see on this slide, about 2 million vaccine injections so far have been done in the world using the Pfizer vaccine. And you can see that there were only 21 confirmed anaphylaxis shocks with no deaths, so that the incidence is about 11 per million infusion, which of course is very reassuring. I am finishing my presentation with this last slide, probably to frighten everyone and to tell you that the story, unfortunately, is not over. The mutation E484K observed in the receptor binding domain, RBD, of the virus results in a considerable decrease in the effectiveness of the neutralizing antibodies, and this might question the vaccine efficacy in the future if we are of the present vaccine efficacy in the future if we are unlucky. So having said that, now I would like to leave the screen for our first speaker, Jean-Philippe Desli, and remind you that there will be a short live discussion at the end of the webinar. Thank you very much and see you in an hour.
Thank you very much for inviting me to this seminar. I'm going to talk about the coronavirus and the SARS-CoV-2 mainly, and the present context. Hope I'll give some science into where we are at the moment and how science can help in developing countermeasures against this virus. So, first of all, we're going to talk about emerging viruses, and I'd like to put this quote of the Surgeon General of the United States back in the 60s, when we were very proud of the achievements, mainly of antibiotics, where the surgeon said that the time has come to close the book on infectious diseases. We have basically wiped out infection in the United States. So as we see, this is far to be true now, and because of the viruses we experience, we see. And before that, I will give few words about where we come from and where we are heading, and in comparison and to the current pandemic. So before the 20th century, we mainly 3 to 4 big infectious diseases were plaguing the world, mainly the plague due to Yersinia pestis, smallpox, which is a DNA virus, the variola major, which was eradicated in the 70s, and the cholera, which is a bacterium and is still also running in some places in the world. Probably there was also influenza virus and RNA virus that we know, the flu. And here on the right, you can see the trace of how RNA viruses were actually very present in the beginning of the modern human history, I would say, with the Egyptian stem showing that this person has a very numb leg, which was probably due to poliovirus at the time. After the 20th century, we had a lot of influenza. Well, during and after we were able actually to record influenza virus pandemics, and 3 important big new viruses came: the human immunodeficiency virus, HIV, hepatitis C in the 90s, and SARS-CoV-2 right now on this millennium. And so the common theme there is that the big pathogens shifted from bacteria and viruses that we were actually unable to detect to RNA viruses, and these are novel viruses. So emerging viruses, and most probably RNA viruses, are a global challenge. We can see here a table on the left where the number of emerging pathogens between the 80s and in 2005 were recorded. As we see here, the majority are coming from viruses, and most of them, the majority, even the majority in these numbers, are RNA viruses. Most are zoonotic viruses, that is, they are coming from animals, and they are called emerging or re-emerging viruses. And the definition for that, of an emerging pathogen, is that the appearance of an unrecognized infection or a previously recognized infection that has expanded into a new ecological niche or geographic zone, and often accompanied by a significant change in pathogenicity. On the right, you can see the map, the current map, well, a few years ago, of the different emerging diseases, newly emerging and re-emerging. You can see that the whole world is concerned by these epidemics, and most of them, the great majority, are RNA viruses, such as dengue, chikungunya, yellow fever, influenza, and now SARS coronavirus number 2. So how to be prepared? To be prepared against these viruses is to address 3 different things. The first one is where the virus come from. They come from the nature, basically, and most of these zoonotic viruses jump from animals to humans. This can occur during the agriculture growth of or cattle or other animals that are in the vicinity of man, but also when we go into places where there's a lot of biodiversity and lots of different viruses, and we expose ourselves to these viruses, for example, by deforestation and the other intrusion of man into these lands. And so the first thing to do is to deal with this pathogen is to observe the biodiversity, understand how it works, what are the different relations between the different animals, and respect and try not to invade or to kill the places, the animals or the places, and generate this expansion of geographic areas. Once a problem is apparent, we have to be ready for these problems, and vaccination comes to mind because it's the perfect protection and prevention method. But it has, as we saw in the current situation, some technological, chronological challenges. That is, a vaccine, you need to know the pathogen before you are actually designing a vaccine. And so we saw that in record time, we could actually have a vaccine against SARS-CoV-2, but it took a year. And during this year, the virus is expanding. And although we were very, very fast, we missed the first year, which is extremely important for the development. Another aspect, and the third one, is that we need drugs, and drugs, I will come to that in my talk. We have very few drugs against RNA viruses, and then they are linked to early diagnostics, and they must be easily available, and so we are missing a lot of drug for these new viruses. But there is a lot of hope, and the answer for all these three steps is that we need science more than ever. We need a lot of scientific knowledge on these three different aspects to be able to deal with future pandemics, avoid them, and when they're there, control them. We need science, and science is different than technology. It's different from engineering. It's different from medicine. We need science to actually feed technology, engineering, and medicines. And science is really done by academic research. This is a very old slide which presents how to anticipate against RNA viruses. I presented this slide in the early years of 2000, when I was actually involved in a big European project called Vizier, where we reasoned that if we study viruses in general and have crystal structures and mechanisms and character, the mutations, the genomes of a family of all families, big families of virus, and have ready all this science that takes a long time, if we had an outbreak somewhere and we identify very quickly the virus, and this can be done really easily by sequencing technology, novel sequencing technologies, we identify viruses and sequence, and then these viruses, most of the time, they resemble viruses that we already know. And so here, for example, this novel virus really looks like a rabies virus, and it must be confirmed by sequencing, and then we can infer biological and structural data very quickly from what we know we have already developed, in that case against rhabdoviruses and rabies virus. And this saves a lot of work. As you will see for the SARS-CoV-2, it was extremely helpful. Coronaviruses, I will talk about three very important aspects of coronavirus biology and molecular biology: how they enter the cell. The entry of the cell can be compared to a key and a lock. The replication machinery, which is the engine, how the enzyme actually do replicate and reproduce the viruses, and also other targets that are assisting all these two other different aspects of the virology, the virus cycle. The classification of coronaviruses, they belong to a very large order, which is called the nidovirus, nidovirales in Latin, that has to deal with the way they replicate in their cell. They encompassed a lot of animal pathogens. And as you see, they were before 2002 and 2003 for the first SARS epidemics. They were mainly animal pathogens. And even now, only 7 coronaviruses are known to cause human diseases. 4 are not really dangerous. These are the HCoV-229E, OC43, NL63, and HKU1. And 2 members of the clade beta are the SARS-CoV-2 1 and 2 and the MERS, which are actually the dangerous pathogens that we know of this family. Why are they called coronaviruses? They derive from their shape and their appearance under the microscope. On panel A, you see the virus, which resemble the virus particle, which has these little spots on the periphery, which were reminiscent of a corona, a crown in Latin, and so that gave the name of the species. The corona are a family of positive stranded RNA virus, one of the largest RNA genomes, which can go up to 32,000 nucleotides, which is really a lot and has a lot of coding capacity. A lot of proteins are coded by these viruses, which are actually giant-like RNA viruses. Before 2003, as I said, and before the appearance of SARS and MERS, they were only known to cause common colds in humans, respiratory diseases that were benign. And the SARS epidemics changed that. So how do they emerge? How do they pass on to humans? Well, they almost all have an origin which goes back to bats. And bats harbor a lot of different coronaviruses. And in some cases, these coronaviruses can go to an intermediate animal, produce an infection, either productive or transient infection, and then sometimes this infection is actually transmitted to man. As we see here, all the different coronaviruses, benign coronaviruses, come from bat and rodents, and all the dangerous coronaviruses, which are below there, SARS-CoV-1, MERS, and SARS-CoV-2, have their origin into bat coronaviruses, as you can see from the sequence, and were ultimately transmitted to man. As we see for SARS coronavirus 2, the present one, we do not know the intermediate host that was probably responsible for the passage from to humans. So back to 2003, SARS coronavirus emerged from bats, went to civets, and these civets, because they were sold on markets for food, jumped to humans and then began a human-to-human transmission, which is actually the hallmark of a pandemic. And it started in China, then went to Hong Kong and Taiwan, Singapore, a flight to Canada, and then there was an outbreak in the world, which was contained as the virus was not really transmissible, but was pretty dangerous, with a case fatality of a fatality case ratio. About 10% of people were actually dying of this virus. The MERS appeared in 2012 and gave several epidemics, peaks here on this this graph, and you see that the epidemic is still ongoing actually. But this virus is much less transmissible. So far, we have 2,500 cases in the world, but the fatality case ratio is pretty significant, with almost 35%. And again, bats were at the origin of the disease, transmitted to men and camels. SARS-CoV-2 appeared in 2019, as we all know, and this is already an old slide. Old slide means it was taken in October, and you can see that the rate of the pandemic is actually incredibly high, as we see that for, now we have 80 million people, 2 million deaths, And only 3 months or 4 months away from this number, we were on this slide, half the number. And it's still counting with no really signs of decrease. So how science can help and will help, and is actually helping a lot right now for the COVID? Well, we have to, the first thing is to really understand how the virus works, because we cannot really work blindly and have only countermeasure. We need drugs and vaccine. And here is an indication of what is the viral life cycle of the virus. So the virus is actually entering the cell into a process called endocytosis, where there's an invagination, binding of the virus, invagination of the membrane, and the virus is traveling into this membrane until it is released by a process that I will describe later, called fusion, releases all the material inside the cell. The viral enzymes are actually generated by translation on the host machinery. The engine here, as I showed in the slide, is synthesized and replicates this RNA, which is later put back into a vesicle exactly the other way, the reverse way of the entry through fusion, and then invagination and release of millions of viral particles upon infection. And these two processes really share a lot of things in common, as I will show in the remaining slides. So the entry is really dependent on these proteins that you see here on the slide, which are called the spike. And the spike is, in fact, 3 proteins coded by the virus, which protrude at the surface of the virus, and they mediate the attachment of the virus. So this is what crystallography first, and then cryo-electron microscopy gave as a picture of the spike, the atomic resolution of how this spike is made. This trimer, so 3 copies of the spike in the center, are actually made of the monomer. A single copy is represented here, and in this copy we have the RBD, which stands for receptor binding domain, which is a domain here which is actually responsible of finding the part of the cell that will help the virus to enter, an N-terminus domain and a center domain. They assemble into this fantastically precise structure. If we see on the right the structure from the top, and we see that we have 3 different binding domains that are able to attach to its receptor. In this case, the receptor is called the ACE2, which is angiotensin converting enzyme type 2. How did that work? So this domain is actually binding to ACE2, but it is not only binding that promotes entry. It's like a key with a lock. We have the RBD, which is binding to ACE2, and then it needs activation, a little bit like when you have the key in the lock, you would put it in the lock, but then you have to turn the key to activate and make the door open. It is exactly what is happening here. Once bound to ACE2, the protein is actually opening up, losing up, and this allows the inversion of the membrane and release of the material inside the cell. So ACE2 is the cellular receptor at the surface of the cell. Its presence and sequence defines which species are infected. Of course, if we have a lock which is not able to receive the key, it won't work. And so it is not only defining the entry process, but also which animal and which cell and which tissue is going to be infected by the virus. That's why the ACE2 being expressed a lot in lungs, arteries, heart, kidney, and intestine, the virus will substantially enter in these cells to provoke pathologies. So how this activation is made is cellular proteases, enzymes, actually bind to the spike and cleave, and how they bind to a very peculiar site which is unique to coronavirus SARS-2, here represented in this orange box. The SARS-CoV-2 has a special sequence which is unique into its family, and this is cleaved by furin-like proteins to promote this entry. So several cleavages are required to achieve membrane fusion, at least 2 cleavages, and the efficiency of the cleavage is actually determining the infection efficiency. And for SARS-CoV-2, this cleavage is actually very efficient because of this increased efficiency site, furin cleavage site here. How is this species specificity determined is shown in this slide, where you have another representation of the spike protein with the RBD, and you can see that the different amino acids that are changed in the RBD or in the protein actually are specific of species. For example, we have rat and rodents that have this specific mutation for 9.3 at this position. It's not a mutation, it's actually the sequence that defines the species. And one can map exactly the different animals that have the different that have receptors binding to these different amino acids. The NTD is also a site of specificity, and many different work now, sequencing work, are devoted to really understanding how amino acids at this position mediate entry. This is a very useful map because whenever we sequence a new variant, we go back to this map and see where this variant is actually prone to emerge and which animal, which cell. If we go further into the definition of molecular atomic definition, here we see what can be determined by cryo-electron microscopy. Here we see that this is a picture of pink and violet of the RBD bound to this receptor. So we see that this surface is like the glue, or the binding surface here is really determining the affinity. If we go further into resolution, we can map this to atomic details, which amino acids are actually bound. If we go further into definition there, we can see precisely which are the amino acids which are binding and giving affinity to the receptor. Here, 2 of the important amino acids that have been detected into the South African variant. For example, this amino acid at position 484 here, and when it is mutated from E to K, it increases the affinity for the receptor. So we can see that an increase in affinity is actually an increase of binding, and so it translates into an increase of infection. And we can model the different mutations here that will actually decrease or increase the affinity. So basic science here is actually absolutely key to predict the efficiency of infection. And of course, because antibodies elicited by the vaccine will map to these precise sites, it also predicts the success, helps to design very good therapeutic antibodies and vaccines. Here is an experiment about the anticipation, how science is going to be very helpful to anticipate what is coming. So in this series of experiments, researchers have taken the first RBD from the first isolate, the Wuhan virus, and they have evolved it in the lab alone, not into an infectious virus, but into mutated forms of RBDs, and they see that they can actually increase the affinity. If we go to the left here, the different curves are titration curves of these novel receptors, and they actually record the mutation. And what they see is that, for example, the mutation 501, which is found in the British variant, is actually increasing at least 2.5-fold the affinity of the receptor. And if you see, for example, that if you addition this mutant 501 to 484, you increase the affinity about 13 times. So it is actually worrisome that, but the prediction of this experiment is absolutely key to anticipate what is possible to come in the future, and really be prepared to incorporate this mutation into vaccine. So what can we go further here? So the people doing this experiment actually took this variant and included into another experiment and redo the process of selection, and they could actually come to variants that have, as you see here, an affinity of 2.5 picomolar between the receptor and the RBD, which is 600-fold higher than the wild type, which is here 1.6 nanomolar. So you can see that we have these variants do not exist yet. The mutations and the variants exist in the laboratory, but this is what we can expect. This is bad news. We probably will see even much more infectious variants coming in the few months. So we really have to get faster than the virus, vaccinate and contain before we can see these variants appearing. This is a table that summarizes what I've just said. We can actually map the different positions here on the residues, see what is actually possible to get in the laboratory, you know, on the intermediate position there, and what we actually observe on the circulating variants there on the bottom where I showed. And we see that some of the mutations have already appeared, like 484, 501, 477, but novel mutation can actually appear at position 380, 58, 460, etc., etc. So these are the more worrisome variants that are expected in the future. In my next piece of work there, I will present what can be done on the replication machinery, which is really the engine, how this engine can be stopped. So this engine is the Rna Polymerase Complex. It is a very sophisticated enzyme which is able to replicate the virus. That is, you see NSP12, the SARS protein, bound to NSP7 and NSP8 into this very sophisticated heterotetramer, bind to the RNA and addition nucleotide at the incredible speed of 600 nucleotide added per second, which is the fastest RNA polymerase known. The coronaviruses have evolved an incredible machinery there to reproduce and replicate their genome. But the good news is that we have experience into controlling this type of enzyme through nucleoside analog that act like a sand in the engine. For example, the remdesivir, favipiravir, molnupiravir, and all other nucleotides are actually able to get into the active site and terminate RNA synthesis. So the good news is that this enzyme is very conserved, so we do not have the problem of variation of the different serotypes, variants, etc., etc. If we find a drug which is acting on one coronavirus, it will be active on every coronavirus. So remdesivir has several problems. I will come back to this, but the drug concept is very interesting because we can actually store it and use it immediately. And if we find a drug which is already available, this will be extremely important in controlling contact cases, initial cases. Here is what we have. It's compared to HIV, for example, where we have 35 different drugs to treat the virus. Here we are at the beginning of drug design for SARS coronavirus. As I said, remdesivir is not an oral treatment, but people are trying to increase the property of the molecule so it could be used as inhaled at least. And it has some problems like toxicity, which is not, selectivity is not great. It has to be used very cautiously. Molnupiravir is another drug which is being developed and right now in clinical trials. We don't know how it's gonna go, but probably it will also be helpful in controlling the viruses. Therapeutic antibodies are being developed, and the big question marks here are there going to be active despite spike mutations? That we don't know. Probably clever design will help, and the vaccine, of course, are actually working pretty well right now, and we have to include the novel mutants and variants in the upcoming vaccines. The good news is that there's an intense effort by pharmaceutical companies to find novel drugs, and novel drugs are slowly showing up, which is a good, very good news for the control of the epidemics. So other targets, other enzymes are being investigated, and this is a summary of the different parts where we could actually act, and where people have been trying to repurpose or position existing drugs. So, for example, hydroxychloroquine has an effect on the fusion of the virus, but unfortunately, this works in the lab, but does not work on patients because all the cells, lung cells compared to cells we use in the lab, they do react differently to hydroxychloroquine, and so it doesn't show any interesting efficacy. We have a lot of other other drugs that have been used and tested. Most of the time, the effects are not great. They are useful, but they are not the miracle drugs we're expecting. We have to be careful that some of the drugs have been used and repositioned without any scientific basis, really. Like lopinavir and ritonavir, a lot of work has been done on that, but it's not really helpful, and there is no scientific basis because the protease of the HIV from where these drugs are derived is not the same as the SARS protease. Oseltamivir, which has some very minor utility in flu, has absolutely no use in SARS coronavirus because the enzyme which is targeted by Tamiflu or oseltamivir is not present in SARS. So we have to be really keep the science serious there. As my last slide is a proposed action map for therapy. What we need first is absolutely, and this was very apparent during the prison crisis, we need absolutely precise, accurate, and fast diagnostics, because without any diagnostics, we will always have patients that come to the hospital too late. So diagnostics are extremely important. We saw also that during the control of the pandemic, it is absolutely required. Once we have an index case and the contacts around this index case, it would be interesting to give a drug as soon as possible to avoid the viral load, which is responsible for firing an inflammation response. And so the infection basically is of two phases. First of all, the development of virus, and the second, inflammation, which is actually causing a lot of damage, because for elderly people and people who are basically more than 65 years old, we have an altered response to interferon, which is actually promoting this escape of virus development and inflammation. And so a successful therapy stands with three types of intervention: diagnostics, early and precise; antiviral drugs at the very beginning of the infection, and then controlling the infection and the cytokine storm that is actually on the late phase of the virus. And we have more and more indication that it is possible to reduce significantly the mortality due to this virus. As a last slide, I'd like to thank people who have been really involved in the lab and in collaboration in my lab. Etienne de Crolly, François Ferrand, Jean-Claude Guillemot, Karine Alvarez, and Nadia Rabat are the PIs from the CNRS and the university they have directed a lot of the different work in my lab. We have a very valuable collaboration with Eric Snyder in the Netherlands, in the Leiden Medical Center, Johan Nijs for antiviral research and evaluation in Belgium, and Olve Pearson in Colorado State University, who spent a year in our lab, and we had a lot of interesting work done there. In collaboration also with a team in Marseille and the IACH, Bruno Canard, Franck Touré, and Geraldine Piotrowski. And I think I stop there, and I thank you for your attention.
I will speak today about the vaccine against COVID-19. A deadly pandemic spread rapidly around the world within few months, resulting in 1.8 million dead in December 2020. This pandemic has induced an economic collapse. How can we get out this unprecedented health crisis since the Spanish flu? The only way to stop the circulation of the virus is to reach herd immunity, that is, protection against contagion of about 60 to 70% of the population. Without vaccination, this can take years with a very significant human cost. The purpose of vaccination is precisely to accelerate this process and to protect vulnerable people against severe infection. It is the reason why many researchers and pharmaceutical companies have entered the race for vaccines using various and sometimes revolutionary technology with the hope to end the pandemic. So the SARS-CoV-2 is an enveloped viruses, coronaviruses, with a single strain RNA genomes originated from bats. The virus is constituted of four main structural proteins: the S-spike proteins that promote entry in cells, which is a very major antigen for the vaccine, the E-envelope protein, the M protein, and the N-nucleocapsid protein. The virus is transmitted mainly by symptomatic patients via respiratory droplets from cough and sneezes. The infecting dose is 1,000 viral particles. It is relatively stable virus with few mutations as compared to the flu virus. If there is no proven variation of virulence, recent mutants more contagious have emerged recently in United Kingdom and South Africa as a new threat. So the spike protein plays a key role in the entry of the virus. The spike displays strong affinity for a transmembrane receptor called ACE2 for angiotensinogen converting enzyme 2, and a serine protease from the cells cuts spike and allows entry of the virus, and the membrane fusion is achieved, and the genome of the virus can replicate freely in the cytoplasm of cells. So it was important for vaccination to make sure that the natural infection induces protective immunity. That's a prerequisite. Most symptomatic patients spontaneously ill in a few days, but about 15% of patients suffer severe acute pneumonia aggravated by cytokine storm, ultimately resulting in death of 1%. Almost all patients produce antibodies, IgA, IgM, and IgG, and develop a protective T-cell immunity which eradicates the virus and prevents further infection. The protection lasts at least for 6 to 8 months. There is very rare documented cases of recurrence and relapse in cured patients, indicating a strong post-COVID protective immunity. This is reinforced by experimental data in infected non-human primates that acquire a strong post-COVID resistance to nasopharyngeal virus challenge with a strong antibody response as seen in patients. So the virus can multiply during natural infection in the upper respiratory tract to induce immune response, IgA, IgG, and IgM. Protective immunity is due to the production of neutralizing antibody and cellular immunity. Epitope pools detect CD4 and CD8 positive T cells in 170% of convalescent patients, respectively. T cell response are focused mainly on the protein spike, but also in other viral protein. So the objectives of the vaccine, anti-COVID vaccine, is one, save life, because we know that 90% of the dead people are often very old people, so elderly, and patients with comorbidity, including diabetes, hypertension, or obesity. The second objective is to stop the circulation of the virus, to reach herd immunity. So herd immunity, it's the final goal of vaccination because it's the only way to stop the pandemic. In a given population, you have a lot of susceptible persons which become infected and which will recover, and in red there are recover people. So the objective is to obtain 60 to 70% of resistant people, which can stop the propagation and circulation of the virus. So one thing is very important thing is the achievement of an accelerated development of anti-COVID virus vaccine. The convention, you have two ways. The conventional development takes 5 or 10 years, and the SARS-CoV-2 development took almost 12 months or 18 months. So the development of a new vaccine usually takes 10 years. First, a preclinical research phase in animals define vaccine target, efficacy, and toxicity. This is followed by a phase 1 and phase 2 on dozens of individuals in which toxicity and immune response are tested. Then phase 3, where the vaccinated people are exposed during a wave epidemic, and we compare the protection of vaccinated people to that of control receiving a placebo. Review of result and approval takes 1-2 years in the conventional development. This is followed by phase 4 with a large-scale production of vaccine and diffusion of to the general population. In the case of COVID, there was important pre-existing data indicating the spike protein as a major target defined by previous work on SARS and MERS epidemics. Beginning in February 2020, the phase 1 to 3 were overlapping. Due to the emergency, phase 3 was finally completed for some vaccine in November 2020. Licensing was obtained in 1 month, and for 3 vaccines, large-scale production and diffusion began in November and December 2020. This is an unprecedented achievement. So let's see about this, the vaccines that we may have. The race to vaccine, we have more than 150 candidates vaccine in progress. Vaccination strategy was either the conventional methodology using inactivated viruses, as for the flu vaccine, live attenuated viruses, purified protein administered with adjuvant, and innovative methodology using DNA or RNA vaccines. So the inactivated and attenuated vaccine are first, the virus for the inactivated virus is very simple. Virus is grown on cell culture and chemically inactivated, for example, by beta-propionolactone. So we have 2 Chinese vaccines and 1 Indian vaccine at the present time used in humans outside Europe. And for the live attenuated vaccine, you have we take the wild strain of virus, and it is weakened by genetic engineering and then injected live in people. And you have one vaccine like that is an American vaccine, the Codagenics vaccine. For the protein subunit vaccine, they are based on the production of the spike protein in recombinant baculovirus grown on insect cells, used with adjuvant or incorporated in virus-like particles without genomes, and displaying spike protein on the surface as is shown on this slide. You have two vaccines this way. The vaccine from Novavax with nanoparticles phospholipid called Matrix M, and vaccine from Sanofi GSK with the spike protein plus adjuvant. Let's go to the viral vector vaccines. They are recombinant spike and or receptor binding domain based vaccine. So we have a replicative vector, and you have a vaccine from Pasteur Merck, a live attenuated antimorbillivirus vaccine harboring the spike gene. It's in phase 1-2. And you have a non-replicative chemically inactivated vector virus in adenovirus, usually, harboring the spike gene. The most advanced is the Oxford AstraZeneca virus. This vaccine has a big advantage because it can be stored at 4 degrees. So let's go to the DNA vaccine. The DNA vaccine consists of plasmid carrying the spike gene whose expression is controlled by a mammalian promoter. The protein is produced in vivo after injection of recombinant plasmid. The advantage of this technology is the ease of large-scale vaccine production in E. coli and the good stability of the plasmid. However, these vaccines often have a low immunogenicity, and they must be administered with nanoparticles delivery techniques to make them more effective. 4 DNA vaccines are currently in phase 1 and 2 of clinical trials. The most promising vaccine at the present time are the RNA vaccine that have benefited from 10 years of intense and innovative research on SARS, but also on the vaccine against Ebola and Zika viruses. Injected in tissue, we must know that a very fragile RNA is quickly destroyed by cellular enzyme. The innovations consist in developing synthetic viral messenger RNA resistant to degradation and packaged in protective lipid nanoparticle, allowing access by fusion to cell cytoplasm. One use synthesize modified RNA of the spike viral gene or an internal fragment encoding for the receptor binding domain. The spike protein is directly translated in cells and presented by dendritic cells to B and T lymphocytes. So you have two vaccines. You have the Pfizer-BioNTech vaccine from US, Germany, and the Moderna vaccine from the US, which are presently on the market. So these vaccines are very interesting because they are so innovative. You have two very important innovations. The first one is the modified synthesized RNA of the spike gene or its RBD domain. To do that, you have a lot of license and innovation. First, to stabilize the RNA, you put anti-reverse cap analogs, pseudo-uridine are incorporated. You can also design replacement of the rare codon by frequent homologous codon, which optimize and facilitate the translation. Altogether, the RNA is an improved stability and increased translation related, if we related that to the translation of messenger RNA from the cells. So the second innovation of the RNA vaccine is the lipid nanoparticle. The lipid nanoparticle are constitute for this vaccine of polyethylene glycol lipids, cationic lipids, helper lipids, and cholesterol. And you see in red the messenger RNA from the virus, from the spike gene, is incorporated and protected by this inverted micellar structure surrounding the mRNA. So this nanoparticle can penetrate by cell fusion in any cells, including dendritic cells presenting to the MHC, and they trigger a very strong immune response. And during the vaccination, the sequence is this one. When you inject the nanoparticles, they penetrate the dendritic cells. The cells can migrate to the lymph node, and then the dendritic cells present to the CD8, CD4, and B cells the antigen and trigger a very strong immune protective response. So the problem of the route of vaccination, currently all the SARS-CoV-2 vaccine candidates in clinical development are administered intramuscularly. Natural infection of respiratory tract induce secretory IgA and serum IgG and IgM response. Intramuscular and intradermal vaccination leads to a strong induction of serum IgG, but not to an induction of mucosal IgA. The lack of IgA often leave an individual vulnerable to infection of the upper respiratory tract. Intranasal vaccination could efficiently induce mucosal IgA response, thereby providing sterilizing immunity in the upper respiratory tract, thus stopping the viral circulation. However, systemic immune response are often lower. That's a big problem, a potential problem with this kind of vaccine. So at the present time, after the clinical trial, we have 9 vaccines in December 2020. 9 vaccines have achieved the phase 3, and 3 are on the market in Europe and US. These vaccines are inactivated vaccine. You have the 2 Chinese vaccine and the Indian vaccine, which are protective at more than 70% on volunteers. You have the unreplicated viral vaccine expressing spike, and they are mainly designed with adenovirus. The only one on the market at the present time is the Oxford AstraZeneca vaccine with a very good protection. And finally, we have 2 RNA vaccine, one from Moderna, US, and the other from BioNTech, Germany, and Pfizer, US. And this vaccine give a very good protection of 95%. And you may have some side effect, but the side effects are always minor. So the result of phase 3 trial show a remarkable efficacy of viral vectors and RNA vaccine, which confer very good protection. They are easy to prepare, but require to be stored at low temperature. RNA vaccine could open a new era in the history of vaccinology because of their efficacy to present adequately the antigen. So what is the strategy of vaccination for the population? First, you have to define priority to save life and to stop viral circulation. To save life, most countries have chosen to start by the high-risk people, to vaccinate high-risk people, more than 65-year-olds, hospitalized, and people with comorbidities. Then comes the healthcare worker and professional exposed to the public, and then the general population, strong incentive without mandatory vaccination. So I want also to insist on the importance of the logistic. How to vaccinate hundreds of millions of people with vaccines requiring 2 injections and store at low temperature, -80 or -20. You need a requirement of vaccination center and a strong involvement of general practitioners, pharmacists, nurses, and other people. You have a necessity of an administrative management, strict administrative management, recording and follow-up of the side effect, fever, redness, local redness, myalgia, arthralgia. And also the big problem is the communication, the pitfall of the vaccination campaign. How to convince the population of health benefit and safety of novel innovative vaccine, and how to restore confidence. Transparency of communication is very important, and fight against misinformation, fake news, and social network, which are so destructive for the confidence. You have several unsolved questions. The first unsolved question is the duration of protection. Do we have to boost or not? We know that benign coronavirus circulating in humans since decades, responsible for rhinitis or cold, induce a 2-3 years protection. So we hope that it will be the same for the post-COVID immunity. You have an uncertainty about the contagiousness of vaccinated people, as underlined this point before, because we don't know yet if the vaccinated people can be infected and how long they can be infected, and so if they vehicle the virus. The best route of administration is also to be defined. Do we have to inject the vaccine by intranasal, intradermal, or intramuscular in order to prevent the circulation of the virus? The contraindication are allergy. You have some problem with allergy, maybe some disease like the autoimmune diseases. Who vaccinated? The last question. Convalescent COVID patient, former asymptomatic or healthy carriers. Are they protected? Do they need to be vaccinated? The question will be quickly solved in the next months. I thank you for your attention.
Bruno Canard is missing here on the video, but
I'm here.
I hope yes, you are here. Good. So
Sorry.
first of all, I would like to thank the 3 speakers. You gave a wonderful talk, and I am sure that the participants will share my opinion. So that's number 1. Do you hear me? Does everyone hear me? Patrick Berche mentions he cannot hear me. Maybe you should open, I don't know, the sound, something. Anyway, then I want to express my gratitude, of course, to Airinspace, who made this webinar possible. And I would like to let everybody know that this webinar will go to the archives of the International Academy of Clinical Hematology. And in case you want a replay, you can get the replay. Now, I got a considerable number of questions. We are late, but I will still try, with the approval of the organizers, to take 10 to 15 minutes and ask questions. However, I had to select because I had too many questions. The first one may be for Jean-Philippe Desli. You frighten apparently at least one participant about cats, and I would add dogs. And do you have any recommendation, because we never speak of that, about the present pandemic and the fact that, I don't know, maybe 10% of the people all over the world have a pet? So do you have any recommendation or nothing special?
Yes, there is a very simple recommendation. Is when you feel sad, when you think that you have the coronavirus, don't go to kiss your cat. Try to avoid to transmit it something, because then you have exchanges. So that's the only precaution. The cats are not as being highly replicative for these viruses. So apparently there is not a big issue here, but precaution implies that when you don't feel well, you don't kiss your grandparents and you don't kiss your cats.
And would it be the same for dogs? Probably.
Dogs apparently are less problematic than cats.
Okay. So, all right. Then I selected, as I said, some questions for Bruno Canard. I'm going directly to probably the important points. In fact, as I said in my introduction, we don't at the moment really have drugs, target drugs, that we're working on and which we believe are going to be probably effective. You mentioned a few drugs within phase 1 and 2 studies, but there is not much publicity about this. Do you have a favorite in mind? Something which is coming that we do not know yet?
Well, there are drugs that are under evaluation right now that I can't really disclose. There will be publication pretty shortly about drugs which are repositioned. I think everything has been done, and there is no miracle drug, but there will be drugs that will be helpful in combining several of those for attenuating the effects and managing the cytokine storm or any inflammatory process that is coming in the late phase, which is very important. As you saw for the dexamethasone, for example, it really is helpful. And there will be other drugs to manage patients. But in terms of having new antiviral drugs that are effective at the very early stage, there are some drugs that are being developed, that are being evaluated, and that will probably be, well, the grail there, you know, the golden standard is an orally available drug that we can actually have before any type of contact, prophylaxis, or contact cases. So we cut the viral load. There are few drugs that are being developed. The present drugs, remdesivir, molnupiravir, and are drugs that have a problem. Remdesivir, the problem is known. It is not orally available, and it has probably mutagenic potential because it's a modified base. So I think it's a very, very first generation drug, which will be abandoned. As you remember the HIV time where AZT was the first drug, it was a poor drug, but it was the only drug at the time. And so it's becoming, you know, it's going to be improved. So we're right now at the phase where there will be, everything has been probably done for the repositioning of the drugs, and the novel drugs will take 6 months to a year to come. But the good thing is that they are targeting conserved enzyme, and so the problem, if we find a good drug, it will be active on any type of variants and things like that. So this is the good news.
Okay, so I'm jumping because we don't have much time. You also, Bruno Canard, frightened everyone with the, the high affinity for the receptor of the new mutants. And you showed us the curves going to the left with a higher and higher affinity. So the question is, and the question in fact could be also for Patrick Berche or for everyone, do we really know that the present messenger RNA vaccines will not protect us against the South African variant? I'm not speaking of the UK variant, but you showed us that the South African variant, in fact, had a very high affinity for the receptor. And so the question is, what about the present vaccine?
Okay, so it's such a rapidly evolving situation that it's amazingly rapid. So even a week ago, I wasn't aware of what's going on. So every day almost, there are papers coming out, preprints. So we have to be careful about preprints because they are not peer reviewed yet, but you can see that when the team has a very long history of good science, we can trust what is in there. And so as soon as, even this morning, I was reading a paper and updating my knowledge on the variants. It seems that not only, and it's not, I'm not trying to frighten or, I'm just trying to interpret what is actually written on these papers. And so what I see, what is clearly stated in this paper is that some of the upcoming variants do have some diminished efficiency for the antibodies elicited by the vaccine or the patient's existing antibodies. So this is a fact, and it's becoming more and more precisely mapped to amino acids. So we are in a hurry right now, in a race of about stopping as much as we can the spread of these variants so we can adapt the vaccine as soon as possible. This is a fact. The second fact is that if you try to predict and anticipate how much better affinity you can get with the RBD and the receptor, there is a lot of improvement in the affinity that can be recorded in the laboratory. And since what we have seen in the laboratory and what is happening with the British variant, the UK and the South African variant, this is really well correlated. So the situation say that examining the data says that they must be there will be probably variants of increased affinity and increased problem, even probably more worrisome than the South African variants. This is what we see from the data. Now we know that nature sometimes plays tricks, and it may not happen, but what we do know today is that it is a possibility.
So
there are two phenomena which are annoying. The first one is that when you treat in vitro with neutralizing antibodies, you are able to push the emergence of variants. So only in vitro, and it's very efficient and very rapid. Second one are the news from Brazil. From Manaus, because apparently we thought that there was a herd immunity a few months ago because more than 70% of the population were infected. And apparently there is a new wave and with a huge mortality linked to a variant. So these two facts combined indicate that there will be a problem.
So you are both of you pessimistic. So Patrick Berche.
I agree for the Brazilian mutant, but for the UK mutant, I think it seems that vaccine might be very efficient still, because you don't see relapse, for example. When you have 60,000 contaminated a day, you must see relapse with the new variant. It is not the case, first. And second, it has been shown that the antibody are still active on the virus. For the South African variant, what is very impressive when you look at this variant is all have the same mutation separately selected. It means something in terms of structural, I think Bruno Canard might speak about that, but it means something that in very different places you have exactly the same and accumulated mutations that change the conformation. In terms of protection, we know that the neutralizing antibody are less protective, but you have many other epitopes on the spike protein which might impede the virus to escape from the immune system. And also you have innate immunity and cellular immunity. So in my opinion, maybe we will have for some variant to use new vaccine, but at the present time we cannot be sure that this variant will change completely the vaccination procedure.
But I had a specific question for Patrick Berche again, along the same lines. Among the existing vaccines today, wouldn't it be reasonable to think that if you take a killed or attenuated viral particle vaccine like the Chinese or the Indian, you might get a larger spread of immunity or antibodies against various epitopes? Would it be, I would say, unreasonable to think that this kind of vaccines might be more effective for the mutants as compared to the RNA existing vaccine of today?
You're right. In the perspective, in my opinion, we will have to use several vaccines. And as you mentioned, the inactivated vaccine, which have all the antigen, might be interesting with a lower protection, 60 to 70, like for the flu vaccine. But that might be interesting. More you have a narrow spectrum, more you may have to face mutants that can change the protection. So I think either the vaccine with the spike protein altogether and the vaccine with the kill virus might be interesting to use on the field. Probably several vaccines will be used and will be complementary as the epidemic proceed.
We have 2 more minutes. Would you say that, and then I have a question for the 3 of you, but just to finish with Patrick Berche, I mean, would you do something which clinically seems not reasonable, 1 shot with vaccine A and 2nd shot not with the vaccine A, but with the Chinese vaccine?
We cannot.
The virus.
I think we cannot do that. The clinical essay trials have been made with the same vaccine. So if you need 2 vaccination, 2 injection, you need to use the same vaccine. That's my answer. We cannot do other way, because otherwise we don't know.
So my last question, I think that's okay. My last question for the 3 of you, and I don't know who is going to answer. You know, people are always frightened. They are frightened of everything, the virus, but also the vaccine. We know that. Not all people, but some. Wouldn't, and especially the messenger RNA. So suppose you get, at the time you get the vaccination with the lipid particles and the messenger RNA inside, and at the same time you get an infection by a retrovirus. And the question, I know that it's not dangerous, but I would like that you confirm that. Suppose that you get the reverse transcriptase of this retrovirus active at the same time? Wouldn't it be possible that the spike mRNA coding sequence goes then directly in the DNA? Is anyone sharing this concern, or is it really totally stupid?
There are more probability to have natural ways and other ways of having problems. The first one is that if you are immunodepressed with HIV, you will have a lower pressure by your immune system, and then the coronavirus will develop for a longer time, and you will select a variant. That's the first thing. Second one is that you can combine viruses. Because you will be exposed to different variants, you will be exposed. You see what happened with employees with the mink. They were almost all infected only by the contact with the mink. So you have such a pandemic that naturally the viruses are going to be everywhere. And so, in fact, we are going, that's my opinion, but we are going to see that once more, we are very resistant, in fact, because we have many possibilities to defend against viruses and bacteria. And that's why we are still here. Humanity is still there despite all those viruses and all those bacteria.
Okay, well, yes, last word, Bruno Canard.
Across evolution, the viruses and their reverse transcriptase and all the systems using reverse transcriptase have evolved to avoid that. That is, for example, HIV is retrotranscribing its genome using a tRNA, and tRNA lysine 3, and it is a very, very specific event precisely to avoid reverse transcription of any type of messenger RNA. So I really don't see why all of a sudden a reverse transcriptase would consider, after these years of evolution to evolve to avoid that, suddenly it would go and retrotranscribe a messenger RNA of the spike, and to do what of it? I mean, it's extremely counter, not logic, you know, it's not logic, and it's probably extremely not frequent. Otherwise, any reverse transcriptase would retrotranscribe all our messenger RNA, and we would be dead. Life wouldn't be possible. So I'm very skeptic about any type of this event appearing.
Okay. So thank you all. We are ending optimistically, and so we should not fear we will survive. You said it's not the end of humanity. Thank you very much. Thank you, Airinspace. And for those of you who want a replay, go ahead. Okay? Bye-bye.
Thank you.